The title of this experiment is cell osmosis. Osmosis is defined as the movement of water down its concentration gradient across a semi-permeable membrane to achieve osmotic equilibrium. When erythrocytes are placed in various concentrations of solution, the osmotic equilibrium will change due to the movement of water molecules. Therefore, when a solution is hypertonic to erythrocyte, the erythrocyte will undergo crenation where the cell shrinks. This is because water diffuses out of the cell and enters the solution to achieve a new osmotic equilibrium. In this experiment, the observation obtained for a hypertonic solution is a colorless supernatant with round bright red sediments at the bottom. The round bright red sediments are crenated red blood cells sink due to centrifuge. When the solution is isotonic to the red blood cell, the red blood cell remains its original shape. This is because no net movement of water molecules has occurred. The osmotic equilibrium is maintained. Finally, when the solution is hypotonic to the cell will undergo hemolysis in which the cell bursts. This is because water from the solution is diffused into the cell to achieve a new osmotic equilibrium. The observation obtained is a red color supernatant with round bright red sediments. The red color supernatant is due to hemoglobin pigments released upon hemolysis of the red blood cells. Therefore, the percentage of hemolysis is calculated by dividing the absorbency of supernatant over the absorbency of supernatant of test tube containing distilled water multiplied by 100%. In the hypotonic solution, a 90% of hemolysis has been recorded, and in isotonic solution, percentage of hemolysis was 1.78%. Theoretically, the percentage of hemolysis for isotonic solution should be 0%. This error occurred due to non-cautious handling of the supernatant when being transferred to measure its absorbency. Next, in the hypotonic solution, a 2.16% of hemolysis has been registered. This is due to cryohemolysis which had occurred. According to the Journal of Membrane Biology, cryohemolysis occurs when human erythrocyte is incubated in a hypertonic medium of 20 to 50 degrees Celsius, subsequently cooling it down to 0 degrees Celsius. Therefore, since this experiment is carried out in an air-conditioned laboratory, our team concludes that cryohemolysis has occurred in the erythrocytes, as the erythrocyte was transferred from a 37 degrees Celsius body temperature to a room of approximately 16 degrees Celsius. Therefore, cryohemolysis has occurred, and that is why the hypertonic solution has a tiny amount of percentage of hemolysis. Next, the control solution has a percentage of hemolysis at 100% as distilled water is hypotonic to the erythrocyte.
In A2 experiment, the control test tube is filled with distilled water. Distilled water is hypotonic to the red blood cell, so water will move into the red blood cell by osmosis down the concentration gradient. In this case, the red blood cell undergoes full hemolysis where the percentage hemolysis is 100%. From the results obtained, 0.45% of sodium chloride is considered isotonic to the red blood cell. In this case, the movement of water molecule from the solution into the cell as well as from the cell into the solution is equal. Therefore, there is no net movement of water molecules. On the other hand, 0.1% of sodium chloride to 0.4% of sodium chloride is considered hypotonic to the red blood cell. In this case, the water molecules will move from the sodium chloride solution into the red blood cell by osmosis down the concentration gradient. This causes the cells to increase in size and swell up. Eventually, the red blood cell undergoes hemolysis where the red blood cell dies. When the red blood cell dies, hemoglobin and intracellular components are released out of the cell into the extracellular space. Upon centrifugation, the supernatant is light red in color. This is due to the fact that the red pigment in color due to the red pigment of hemoglobin. There is a small portion of cells left remaining at the bottom of the test tube.
Test tube containing 0.5% of sodium chloride concentration to 0.55% of sodium chloride concentration. The percentage of hemolysis plunges, and from 0.55% of sodium chloride concentration onwards, the percentage of hemolysis decreases steadily. Overall, the increase in sodium chloride concentration will decrease the percentage hemolysis. This can be explained by the intracellular fluid of the RBC diffuses out to the environment. This causes the RBC to shrink. No osmotic pressure is generated to push the hemoglobin out of the cell. So, the supernatant layer of the test tube is colorless. What we observe at the center of the test tube is red blood cells that remain intact and red blood cell that dies. Thank you.
Start.
Part B. For test tube 1 and 3, 300 milliosmol of glucose and 300 milliosmol of sodium chloride are isotonic to red blood cell. Therefore, no net water movement occur. For test tube 2, 300 milliosmol of sucrose is hypertonic to red blood cell. Therefore, water moves out of the red blood cell and causes secretion of red blood cells. However, there is a low percentage of hemolysis which is caused by cryohemolysis, whereby a drop in temperature from the 27 degrees Celsius of human body temperature to the 16 degrees Celsius of room temperature causes the red blood cells to hemolyze. For test tube 8, distilled water is hypertonic to the red blood cells. Water molecules move into the red blood cells down the concentration gradients by osmosis through the plasma membrane.
Yeah.
Net ion flow of water molecules into the red blood cells continue until hemolysis occurs. For the conclusion of this experiment, the concentration of sodium chloride that is isotonic to the red blood cell is 0.45%. Next, tonicity is dependent on the solution's osmolarity and its non-penetrating solids. When the solution is hypotonic to the red blood cell, the cell will undergo chelation. And when the solution is isotonic to the cell, there will be no net movement of water molecules. And finally, hemolysis occurs when the solution is hypotonic to the red blood cell.